talk will be given by Michael Good and is about the development of a semi-synthetic whole parasite vaccine. Thank you.
Thank you very much, Ursula and Sophie, and thanks to the organisers for inviting me to speak. I'm going to talk about whole parasite vaccines for blood stage and why we think that's a way to go, and then describe a few of the challenges that we have with that approach and how we're trying to overcome those. So this slide here is one that from Alyssa Barry that I often refer to, summarising the problems with antigenic polymorphism in all the, or most of the known vaccine candidates for malaria, looking at the number of haplotypes versus the number of sequence available in the database at that time. They're all polymorphic, which is a huge challenge, and one of the ways to overcome that is obviously to combine them. The exceptions, of course, are maybe E140 when we find out what that is, and also the NANP repeat of the circumsporozoite protein, where an unattainably high antibody response is required and isn't achieved by infection. So the real argument, one of the arguments for a whole parasite vaccine for the blood stages is that a whole parasite infection can induce immunity. And this has been done in many groups, including our own many years ago with this model in Vincii, where you infect the mouse, after a week you treat it and you infect it again, and it's protected, and that protection persists. Now, when we did a similar experiment many years ago now in humans, we infected volunteers, the work of David Pombo et al., when we infected volunteers with falciparum, treated them after a week so they weren't sick, and then measured their immune responses. We saw a strong proliferative and interferon response and a nitric oxide synthase response. There was very low antibody responses in these volunteers. So that gave us more rationale to follow this approach. But of course, infecting and treating a week later is never going to be a vaccine approach. You have to infect and treat on the same day, or use a different approach with whole parasites altogether. And that's what I'm going to talk about just briefly. So immunity following infection attenuated in vivo on day one. Now here's some preliminary results which we have using doxycycline, which is a delayed death drug, as you will all be aware, treatment on the day of infection. And the doxy is then given every day, and that's a week of treatment. And then the mouse is rested, and it may get a second infection drug cure or a third, and it finally will be challenged. Now, doxy, because it is a delayed death drug, allows the parasite to go through 2 cycles of replication. So the immune system is exposed to parasite for a slightly longer period than it would be for some other drugs which are used to treat an infection. Now, when you then look at the parasite persistence during each cycle, whether you give the mice a slightly higher dose, 10^7 parasites or 10^6, after 2 cycles in vivo, the parasites are, of course, cleared by the drug. And you come back and challenge those mice sometime later, and in different mice, and this is with bulbs and black mice, with Shabodi, here is the infection. Whether they've given 10^6 or 10^7, theirs were given that with doxy. These mice are strongly protected. So here you've got potentially a vaccine strategy. At least it's the beginnings of one for a whole parasite. Now, you can do that even with a single cycle of infection and drug cure of PULEI. And here it's on the scale's broken, but these mice here got 10^6 parasites, and given 1 cycle of doxy for a week, and these mice got 10^7, and following challenge weeks later, they are strongly protected. So that's an in vivo attenuation at day 0, and this is an approach that we are now following up with human volunteers, and those experiments are currently underway. But there are, of course, other ways to attenuate the parasite at day 0. You can do it in vitro. We published some of this a little while ago, but I'd like to show you some data from Amber Raja, who was just finished a PhD in the lab on Plasmodium uelei. But the strategy here is to take parasites and harvest the ring stages, or you can do it with the trophs if you wish, and treat them with a drug such as centanamycin or tofuromycin A which binds the DNA and prevents parasite replication. You wash the unbound drug out, you count and inject the parasites into the mouse. And when you do that for either 1, 2 or 3 cycles, depending on the parasite, you can get very strong protection. In this particular study here, what Amber has done is to immunise mice with PULEI and give them a sporozoite challenge via mosquito. These mice do not have liver stage immunity, but we're looking at the blood stage here. And these mice who are immunised by attenuated, intact, parasitised red cells are now very strongly protected compared to the control animals. When you look at the immune response early on after immunisation, prior to challenge, of course, what you see, interestingly, well, from us it's interesting at least, is you can see activation. This is after the first, second or third immunisation strategy, procedure. You can see minimal activation looking at CD11A markers in CD4 cells after the second vaccination, non-significant in CD4s after the first or the third. But interestingly, in the CD8 cells, you see early activation after the first, the second and the third immunisation of these mice. This is using the method that John Hardy, I believe, pioneered to look at early T-cell activation. So it looks like the CD8 cells are being activated and maybe to a limited degree the CD4s. This is in peripheral blood though. But when you deplete the cells, the CD4s and the CD8s, what you see is if you deplete over on the left there, the CD4 cells, they're the ones in the orange colour here, the mice lose their immunity. This is measuring parasitemia. If you deplete the CD4s and the CD8s, they still lose their immunity. If you deplete the CD8s alone in green here, they do not lose their immunity. But interestingly, if you deplete the CD4s and the CD8s, the mice all survive at a high parasitemia. So we believe the CD8s are contributing to pathology in these mice, and the activation that we saw previously in the previous slide of CD8s are cells which are not helpful to the animal but are detrimental. When you then look at the memory response of these mice, Amber went out to 9 months. This is looking at the 6 months. So this particular slide here, these mice are immune after vaccination and challenged 6 months later. Vaccinated with 3 doses of vaccine. And looking at the T and B cell memory responses in vitro, you see some activation of T cells comparing normal red blood cells to parasitised red blood cells in vitro versus splenic T cells. Stronger activation of the B cells, looking at pre-challenge and post-challenge, and a boost in the antibody response after 7 days, indicative of a memory response at 9 months post-vaccination. Now, that's all very fine, and we are actually pursuing this approach in a number of human studies, but there are logistical problems with developing attenuated blood stage vaccines, whether they're attenuated in vivo or attenuated in vitro. That is, and I haven't shown you all this data, it is published, so the need for red cell membranes to remain intact. The parasite has to be intact, the red cell has to be intact. The potential for inducing anti-red blood cell antibodies, particularly as the red cell dose increases, that can happen, even if you use, for example, Rh negative, O negative blood. There are ethical issues around giving red cells to some individuals, certainly, and there are difficulties of cryopreservation. All these issues, I think, can be addressed to a limit or to a large degree, and we're trying to do that, but they are issues nevertheless. So to overcome some of these issues, we are currently looking at a different approach altogether, and that is to use parasite antigens from the entire organism, from which we will strip off the red cell membranes and enclose the whole parasite antigens inside a liposome here. And on the surface of a liposome, we're anchoring mannose with different linking groups to target the liposome with parasite inside to dendritic cells, antigen-presenting cells. So we've done this with a number of different mannose and a number of different linkers to compare them. It's a very empirical sort of research, I believe. The ones that are looking more interesting, I'll show you some data, are this mannose here, which has this linker and the dipalmitic acid C16 tail, and this one down here with a PEG and a single C16 lipid tail. This is just showing what these liposomes look like when they're stained with different dyes for the liposome, the parasite, and the and mannose, and all put together here, this is just what they look like. When you look at the uptake of these liposomes by either mouse or human dendritic cells, you certainly see significant uptake of these monosyllated liposomes, and F3 and F4 seem to be the best in mouse and in human, and they're the ones that we are most interested in. Looking at the early activation following vaccination, now mice have been vaccinated with these liposomes. This is the work of Ashwin Gidam, postdoc, now in the lab. And these mice are vaccinated. Again, we see minimal and non-significant activation of CD4 T cells in peripheral blood, but significant activation again of the CD8 T cells from the peripheral blood, which is again of interest to us. When you look at the spleen cell proliferative response, whether you make a vaccine in Chabody or in the left here or in Uwelii, you're seeing proliferation of mice vaccinated with the F3 and F4 liposomes, the monosyllated liposomes here in response to parasites in vitro. And these T cells, CD4 cells, make interferon gamma. And finally, just showing you some of the protection data, because this has just recently been published, we're seeing here the mice vaccinated with the monosyllated liposomes, either F3 or F4, are completely protected against a Plasmodium Chabody challenge, and the parasitemia is reduced, significantly reduced in these mice. This is really early days in this strategy for us, and we are looking for ways to experiment with the delivery and the dose, etc. But we do believe that this has certain benefits over intact infected red cells as vaccines. But we'll have to wait and see how these ultimately compare, and we do plan to actually test these in human volunteers down in the near future. So in summary, malaria parasites attenuated in vivo either at day 0 or day 7, which is more classical, or in vitro at day 0 can induce protective immunity. There are logistical issues, and liposomes expressing mannose may be one way to overcome them and develop a vaccine which is safe, has very few, if any, ethical issues associated with it, and can be stored as a powder, dried as a powder, and hydrated at the time of inoculation. These liposomes can induce complete protection in Chabody and in Plasmodium uwelii in the experiments that I've shown you. There are many people on this slide, but I want to acknowledge particularly the first 4 here. Ashwin did work on the liposomes. Amber, who's now with John Adams, who did a PhD with me on the uwelii model. Leanne, who's looking at the doxy, and now applying that with Danielle in a human study. I also like to acknowledge over here Stephen Hoffman, with overall strategy suggestions and provision of sporozoites for our studies, and his group, and Ian Coburn at the John Curtin School of Medical Research in Canberra, who was very helpful and instrumental in doing some of the sporozoite challenges studies that I didn't show you all the data for. And we have a number of acknowledgements to thank, and no disclaimers that I'm aware of, but thank you.
Questions?
Got it easy. Steve. Okay.
So can you go over again from, most of us when we think about blood stage vaccines think about variability of the antigens and so on, and even as it might relate to T cells. So what do you know, or what do we know about the lack of requirement for that in this system?
So the published data, as many of you would be aware, in human trials are really around MSP1, MSP142, and AMA1, where there was no protection in phase 2 studies. There is protection in those models with homologous challenge, and a lot of work in animals led to those studies. I think those are immunodominant antigens, and I think other antigens which are under development, such as RH5, may be much more exciting. These are essentially highly conserved but cryptic, so they're not normally immunogenic as a result of infection, therefore variants haven't been selected. So I think that's I think the main reason, that, and the second reason being the inability to develop a very high immune response to many of these antigens, is why it's been very difficult to develop a blood stage vaccine with a subunit approach. Now, that's not to say that if we can't get smarter or use E140 or something else and put a whole lot of them together, it might work. But the easiest way to put them all together, I think, is to use the whole parasite. And we know that infection with the whole parasite does lead to very, very good immunity. The question is, why don't people develop immunity following an infection? And I think that's the main question to address. And I think one of the reasons for that is because the immune system undergoes significant rearrangement after infection, as we heard this morning and as we all know, and there's good evidence that there's apoptosis of T cells following a malaria infection. I believe if you can then overcome that, and one way to do that might be with a very low dose, a parasite approach, where we know you don't get apoptosis, you can activate your T cells. If those T cells are regulated in some way, I believe they could induce a safe and protective immune response against all strains. That's the overall sort of strategy, but it hasn't been tested yet, but that's what we're hoping to do.
One very specific question. Do you know with the mannose that you actually require the mannose receptor for taking up the liposome, or does it work?
We know this. Thank you. No, the short answer is no. We don't know you require the mannose receptor. We know it's not taken up without the mannose on the surface of the liposome. And we know it is taken up by dendritic cells, CD11C dendritic cells in human and in mouse with the mannose on it.
Okay. And second question: Have you done heterologous challenges?
Yes.
In
Sorry.
relation to
No,
this?
we have, and I didn't think with all that data I'd have time to show it all, but there is good 100% survival with heterologous challenge following ULEI vaccination with Shaboti. Yep.
Thank you.